Thank you very much. So it's a pleasure to be here. And what we will do in this talk is to primarily cover one of the main services in the EBRAINS research infrastructure. Thank you. So in the first part of the talk, I will focus primarily on the sharing and how to find data, and then I will move on to demonstrate more on the use of the data in the system. So, general background statement. We are in the middle of a revolution in science with the recent possibility to make large amounts of data widely available in combination with computing resources. This is a big and recent change. So we started with journals in the 17th century, controversial. People didn't want to publish. 20th century, peer review. Well, that's very accepted nowadays. 21st century, just for a few decades, sharing of data underlying the publication being the norm. So we are in a competitive and changing landscape, and for these reasons, tools and data are drivers for science. We're asking new questions because we have access to data and we have access to more resources for analysis. We have the open science trend, which aims to remove barriers for sharing. And we have a crisis, which is very much emphasized by leading journals on reproducibility and replicability, simply because very often there's no access to the data and not sufficiently well described procedures for what's being done. So the challenge is to deal with that somehow, and that's about being able to integrate data of heterogeneous origin. Integrating information is difficult just based on papers. You need to go into what's behind the papers. Then to facilitate reuse and promote reproducible, replicable science. So in this endeavor, you have the data providers that are coming in with data at many different levels, from gene molecule level and through the neurons and the systems and the networks to the whole brain. And those who are using the data consumers, and we are usually on both sides. So how do we make this change possible? So the EBRAINS Data and Knowledge Services contributes to this by providing a user-driven data sharing, data management platform. It has three main components. It has a knowledge graph, which is critical for organizing all the information. It has certain standards for the language being used, or the metadata, OpenMINDS, and it has services for curation. And we'll try to look at what this consists of. So here is actually the knowledge graph. You can look at it on the web. The content is organized in this three-dimensional, or actually four-dimensional graph, because it's changing over time with the metadata, with tools, pointing to which data can be analyzed with what kind of tool, where the data are located, and all the relations among them. So that's the metadata management system, which is a very good technology because it is flexible. It can be expanded. It can grow, just like our field will have to adapt to future needs. That is what we're building this on top of. But for this to make sense, we need a framework for our metadata, because if we are using lots of different terms without explaining how they relate, we will get just as confused as if you search for a given word in PubMed and you try to find papers that are describing the same. The OpenMINDS metadata framework, actually the leader of that work, Bjaalie, is here. She'll be in the next talk. That is collecting a number of metadata models, control vocabularies, bringing in ontologies, making sure that we create this kind of language, a more standardized language, which is critical. Otherwise, it's going to be very difficult to understand what the content is. And it's been done in a very international context, which is also very critical, because there's no meaning in having isolated standards. That doesn't make any sense. Then we have a curation workflow. This is a critical quality assurance process. We take the trouble of controlling some steps on what is being shared, in principle shown on the lower diagram. It is a process where those who contribute data actually get some help in making sure that their files are organized in a way that others can interpret, and that they are adopting the metadata standards correctly. And then there's an upload to the storage system where the data will be available. So in this way, we can increase the level of fairness, findable, accessible, interoperable, reusable data, going from left to right. The more information we have, the more fair it will be. Journals will require it, funders require it. So this is life. This is here to stay. And the more you go up, the better it will be. But of course, you need to pass some thresholds, and that's fully possible in this system. So the service for sharing goes through the curation. The services for finding data are both graphical user interfaces, search interfaces, as well as programmatic access routes that goes directly to the knowledge graph. So just having a graph is not sufficient. You need the tools on top that allows you to search that graph, which we have. So what are the reasons, what are the arguments? Maybe you want to make your data available with a journal publication. Would this service be of interest? Yes, it is, because we have set up mechanisms aligned with, for example, Nature Scientific Data and other journals, which gives you special tracks for publishing data descriptors and publishing your data, regardless of essentially which journal it is. Maybe your data is of a foundational nature. It is something that obviously can be used by many. Then, with the curation, with the metadata standards and the advanced discoverability, it's a good place to have those data, or to make the data discoverable if they are located elsewhere as well. This is very important in relation to international collaborations. Also, having the metadata in the system pointing to data elsewhere is an opportunity. And of course, then to find and combine data is really the ultimate way forwards. And there are many using this now from outside of where we are coming from. These are just 3 examples from very different places in the world, bringing in data into this system. And I'd like to emphasize that we can take everything from non-identifiable data from humans, meaning that no one, not even the researcher, can identify where they're coming from, that they can be shared directly. And of course, all kinds of animal data. They may have an embargo, meaning that there is a waiting time before they go and are available to people, but you can find them. Then we have restricted access for strongly pseudonymized, de-identified data, where you can get access, not too difficult, but there are certain rules. You're tracked and you're logged. And then finally there's controlled access, very, very tight control, when there's most of the human data falls into that category, requiring very tight control, which is a system we don't have at the moment, but will come next year from our side. And there are several European services that deals with that as well, where we want to have the metadata for those data in this system. This is built by many teams across Europe, and we'll continue then. And we can also say that if you want to hear more about it, there's a Nature webcast, Making Small Data Big webcast. Nature will give you it in one go that summarizes this. And then Maja will take over. So now we've gone from left to right, organizing data model software into a knowledge graph, and then finding and using it.
Thank you very much. So, in the EBRAINS infrastructure, as you've seen so far, we have data sets and models. It's data set about the human brain, of course, but also rat and mice data sets, and computational models. Those, as Jan explained, contain rich metadata that are organized in the knowledge graph. We have a set of reference atlases you can use, and then we also have dedicated tools that are linked to those atlases for spatial registration and analysis. And using those tools, we can make complete workflow. And I will give you some example use cases, for example, in electrophysiology, cell counting, or track tracing. And my colleagues Timo and Luba will show you more about the tools you can use with their human atlases. By the way, I want to point out that we released the 4th version of the rat atlas, version 4, with nice delineations now of the cortex and several subcortical structures. And of course, we have many data sets linked to those atlases. So on the infrastructure, you can find different services. You have the data knowledge service that Jan just showed you about. You have atlas services, simulation, brain-inspired technologies, medical analytics, and general service for community. So if you go to the atlas services, you could find tools like Quickney and Visualign. Those are typically used for rat and mouse data. And they allow you to deform the 3D atlas and find the position of your experimental images. Those tools are compatible with any cutting angles, and you have extensive documentation in the form of reader docs. But also we prepared some tutorial videos you can find in the INCF training space to learn how to use those tools. And the registration to Atlas is a nice first step to do Atlas-based analysis. So, for example, if you want to quantify the numbers and the distribution of cells in your material, and I will give you some examples of dataset you can find in a knowledge graph. So here is a brain-wide quantification of parvalbumin positive neurons done in the rat. All the images were anchored to the atlas, so they have atlas registration. And this allowed us to extract all the positions of the interneurons and transform them to point clouds, having thereby distributions like this. And then we used the Util software to get numbers and quantify those parvalbumin cells in the different regions, and the regions are coded by the colors. So each region is represented by a different color. In the knowledge graph, you will also find similar data about mouse, so you can do comparison. And here, the same way, the data was registered to Atlas and extracted, so you get numbers and can do cross-species comparisons. Another example is connectivity mapping. For example, here, colleagues of ours, Toko and Trygve looked at the impact of a gene NR2F1 in the connectivity between the cortex and the pons regions. And they mapped those connectivity relations both in wild type and conditional knockouts. After registration to Atlas, they used the Localism tool to make point annotations. Here you can place your own annotations and do a quantification of the different connections. And they could show here that in the knockout you have an increase connectivity from the cortex to the pons, but further down in the network, the numbers of projection were decreased. Another example with collaborator in Trondheim by the Vitter Lab, also doing track tracing studies in the orbitofrontal cortex, and here as well use the same approach to extract whole brain cortical mapping of the different regions. And a third example I wanted to give you is that you can use those tools also to look at specificities in electrophysiology dataset. For example, you'll find this dataset in a knowledge graph where they have done striatal recordings of the striatal cholinergic interneurons. And if you go here in this card to related resources, you can see that it has been used to make a model. And then you can access the card for this model as well. So you have access to both the data and the model that have been used to make the model. Another example is from our collaborators in the Penards lab, where they do tetrode multi-area recordings from visual and somatosensory cortices in rat, and they combine the precise location of their tetrodes to the electrophysiological recording, allowing them to really get precise location information in those perirenal cortex regions. So I invite you all to search the knowledge graph. You can use the tools we are using. They are shared. You can use it on your own data, or you can use them on the data we share in the portal. We also have community pages, so you are welcome to join us there. We are happy that we have many users in the whole world, actually, here. And a number of publications coming up now that are using our tools. So thank you very much.